Bismillahir Rahmanir Rahim. Assalamu alaikum, my dear students. I'm your instructor of microbiology, Dr. Aisha Rashid. If you do, people remember we were discussing the microbial diseases. Under the series of microbial diseases, today's discussion will come under lecture number 24. So today's disease which we are going to discuss, the first one is malaria. Most of people are very well familiar and known what is the malaria. So first of all, the introduction of malaria. Malaria is probably one of the oldest disease known to the mankind that had a profound impact on our history, on our community. If we see, there are a number of malarial cases every year all over the globe. It is huge social, economical, and health problem, especially in underdeveloped countries and particularly in the tropical areas. Malaria is a vector-borne infectious disease which is caused by a single agent or single microbe which is called as protozoan parasite, and the genus of it is Plasmodium. The malaria is transmitted from one person to the other person by the bite of female mosquito. Here you can see the female Anopheles mosquito transmit the malarial disease. Basically, its trunks or spikes which carry the infection from one infected person to the other one. What causes the malaria? Malaria is caused by a parasite which is called as Plasmodium, which is transmitted via the bite of female Anopheles mosquito. In human body, the parasite multiplies in the liver, and then from liver it infects all over the blood in the body. Then the symptoms appear. The transmission of the malaria do not occur less than 16 degrees C and greater than 33 degrees C. That is why we are saying its spread is mostly in the tropical areas. Malaria kills more people than that of AIDS. It is mostly caused disease and transmitted from one person to another. So its spread rate is very high. Malaria kills 1 year in 1 year what AIDS kills in 15 years. So its ratio is 15 times greater than that of AIDS. For every death due to HIV, there are about 50 deaths due to the malaria. To add the problem in the problem is the increasing drug resistance to the established drug. So the major factor which we have seen why this much spread is happening, because Plasmodium is continuously changing its genetic makeup or its behavior, and it's becoming resistant towards the drugs. That is why it's not cured. So it is seen that it is more spreadable disease than that of AIDS. Broad clinical manifestations of malaria. The clinical symptoms or clinical indications of malaria includes fever, high-grade fever is there, sweating, anemia, there is iron deficiency in the blood, then splenomegaly, which is called as enlarged spleen, then irritability, patient may go in coma, then a retinal hemorrhage may be there, respiratory distress syndrome, RDS, can happen as well. Now the laboratory diagnosis, how the malaria is diagnosed. Laboratory diagnosis of the malaria can be made through microscopic examination of very thick and very thin blood smears. Blood smears are firstly made on the glass slide, then this glass slide is observed under the microscope and for the causative agent. Thick blood smears are more sensitive in detecting the malarial parasites because the blood is more concentrated, allowing for the greater volume of the blood to be examined. However, thick smears are more difficult to read. Now, blood collected with the sterile technique. For this testing of the malaria, it is very much important that the sterile technique for blood collection must be adopted. Mean every tool must be sterile. So there may be no, you can say no agent or no other spread of the disease. Here is the diagrammatic description of making a thick smear, just like for other blood tests, how the thick smear looks like. And under the microscope or after preparing the slide, thick smear look like this. Now when this thick smear is observed under the microscope, then how microscopy appears. Malaria parasite can be identified by examining under the microscope a drop of patient's blood spread out as a blood smear on a microscope slide, as we have seen in the previous slides. Prior to the examination, the specimen is stained, means most often with the Giemsa stain, to give the parasite a distinct color and appearance. That's why it can easily be seen under the microscope. This technique remains the gold standard for the laboratory confirmation of the malaria. Here you can see the results of a thick smear and thin smear. Blood smear which is stained with the Giemsa stain. Here you can see in the maroon or red color, there are some maroon and red color. Here are some parasites, that is the Plasmodium. Now, epidemiology. I have already told you what is epidemiology. Spread of any disease in any community is called as epidemiology. The study of spread of a disease in a community is called as epidemiology. So malaria continues to be one of the most important and devastating infectious disease in developing areas of the world. Worldwide, over 40% of the population lives in the area where the malaria transmission occurs. That is, parts of Africa are mostly infected. There are some parts of Asia which are also affected by the malaria every year. The Middle East, the Central and South America, then Hispaniola and Oceania. These all are the areas where its epidemiology is high. Now, how the malaria is controlled? Elimination of mosquito breeding places, like every year it is said that no stagnant water should be there, every water must be drained, use mosquito repellents, and every remedy must be adopted. National improvements on the health and hygiene, use of mosquito nets treated with the pyrethrin, clothing with the sleeves, no naked sleeves or legs should be there, and long trousers must be put on. Use of mosquito repellents. This was about the third in one infectious disease, malaria, we have discussed. The fourth and important one, we will go towards the gastrointestinal infection. And among gastrointestinal infection, first we will go to the overview. What are the gastrointestinal infection? Most infections of the GIT are acquired to the contaminated food, which we say as food poisoning. Food infection, bacteria like cholera, E. coli, Salmonella, Shigella, viruses like enterovirus, rotavirus, hepatitis A, E, parasites like G. lambelia. These are the all the infectious agent which are present in infected food and can cause the disease. Then food intoxication include SRES, B. cereus, CIA, perfringens, and then Plasmodium botulinum. These are the intoxication agents which are present in the food. Now, what are the symptoms of these gastrointestinal infection? The most common symptom is the first abdominal pain, very severe cramps or pain in the abdomen, and it may lead towards the diarrhea, which can be life-threatening as well. Vomiting and high-grade fever can also be there. Now, the factors responsible for the spread of these food infections or gastrointestinal infection. First of all, age is very important factor which is considerable when we talk about the gastrointestinal infection. Like the children are most susceptible towards these infection. We have seen the children under 1 year or 1 to 5 year, they mostly suffer from the diarrhea. Then personal hygiene meant a lot. Personal hygiene meant a lot. Sorry for interruption. Wait a moment, dear. Personal hygiene meant a lot. Sorry. Now the factors responsible for its spread. Age, we have discussed age, then personal hygiene. Hygienic factors are very much important. Food must be very hygienic, your environment, water, that must be hygienic and contamination free. Open air defecation is also there, which is responsible for that. Then food at roadside or stalls, that is strictly prohibited, that has the much chances of you can see uh, infection. Really sorry for interruption. Okay, uncovered food. If food is at roadside or food is at home, whatever, the food must be covered and hygienic. Now there is vibro, vibrate, or vibratory motility. Vibrio cholera is the causative agent of this gastrointestinal infection and mostly diarrhea. It is present in the surface water worldwide. Now about Vibrio cholera, we will discuss various, you can see the headings, like we will discuss the morphology of the Vibrio cholera, growth characteristics, biochemical reaction, resistance, and then pathogenicity and virulence, epidemiology, diseases caused by this Vibrio cholera, then laboratory diagnosis, and lastly, we will see the treatment of Vibrio cholera. Now the morphology. Vibrio cholera. It is short, curved, comma-shaped bacillus bacteria. Bacillus, you know, which is rod shape or oval, somewhat curved shape. Like you can see, you can say it is just like Sammy seed. It is just like a Sammy seed. So we say that this is curved or comma-shaped bacillus bacteria. Single polar flagella. It has only one flagella which is present on the one side or one edge. That is why we say this is single polar flagella. Then this is a gram-negative bacteria. वालेकुम अस्सलाम, WhatsApp पे नंबर पे भेजना है। हाँ जी, I am on recording and class, please जल्दी से भेज दें। Class recording. Okay, here are the, you can see the shape of this one. You can see this is bacillus, curved bacillus, and this is one flagella present at one side. This is the most clear picture. Uh, this is small curved or semi-curved bacillus with the one flagella. Now the growth characteristics. This is aerobic. This one is aerobic. Aerobic, you know, which needs, you can see oxygen for its growth. This is color. It forms colorless colonies on the Mekongiki agar or Mekongiki broth. We have seen the media. So this is the culture media on which it formed the colorless colonies. The special media which are used for its growth is the transport media, enriched media, and plating media. Now the biochemical reaction by which you can see uh, it can be detected. So cholera red reaction is used for its diagnosis. Then fragments of the glucose or any other sugar like mannitol, maltose, mannose or sucrose is taken, and then the sample is added in that one sugar. Then it will produce indole, which gives the red color. With indole, it will produce red color. This is positive indole test. You can see somewhat purple or reddish is there. This is negative indole test. If there is no bacteria with glucose, no color change will happen. And if there is bacteria, plasmodium, cholera, this bacteria of gastrointestinal infection, it will give you positive color with the indication of change in the color red to or purple. Now the resistance. Resistance to cold. Refrigerated food may carry the Vibro. So that is why frozen food are mostly prohibited and said that no use of frozen food should be there. So frozen foods are strictly prohibited. Then resistant and do not serve at room temperature and heating. Do not survive at room temperature and heating. Then it is susceptible to common drugs like doxycycline. It is susceptible towards the doxycycline. Now the cholera, acute diarrheal illness. It can cause the acute diarrheal illness, or it can cause the painless or rice water like some watery stools. Some watery stool, appearance of stool is rice water like. Then copious vomiting may be there. Fluid loss and dehydration can happen due to this stool and vomiting. Due to two toxins released by the bacteria in the intestine, these toxins are named as toxin A and toxin B. It causes the pouring of the water from body into the intestine. So all body water is poured into the intestine, and from intestine it is lost by the stool. So it will result in the severe dehydration. Then extensive water depletion can happen as a result. Epidemiology. Epidemiology, the study of spread in the community, I have told you earlier. Source. The infected humans are the source of spread. When the infected human stools or contaminated food are there, they can spread the disease very easily. Mode of transmission is contaminated water and contaminated food. Cholera spreads as a sporadic or epidemic or pandemic. And we will see these terminologies: what is sporadic, what is epidemic, or what is pandemic. Large movement of people such as drug during fairs, festivals is associated with the cholera spread. So wherever the large gathering or food sharing is there, it can be the spread, lead towards the spread of this cholera. Now, what is epidemic? Suddenly, many cases are seen or reported over a large area. This terminology is referred as epidemic. And what is pandemic? Cases seen over a long period of time, over countries or over a large area of globe. Till 19th century, cholera was mainly confined to India. Many area of Ganga or Bengal. These were the most infected areas of cholera in 19th century. Total 7 pandemics have been identified till yet, and this seventh pandemic was with the cholera. Here you can see the picture when the in 19th century there was the pandemic of cholera. Now, lab diagnosis, we have seen how it is diagnosed. Sample of the stool is taken before administering any type of antibiotic. Stool sample is taken and examined in the lab for the cholera. Use transport medium is used for its diagnosis. Dark field microscopy can be done for dating motility. Here you can see dark field microscopy where the background is dark and the organism appears as bright. Plated on the TCB's media, then slide agglutination test is there, and then stirring test. This is the detail of the test, how the test is performed in the lab. First, it is plated on a specific media, which is called as TCBS media. Then slide agglutination or aggluting agent is added. Then stirring is performed and results are seen. Now, what is the treatment of this cholera? More important is the replacement of the lost fluid, rehydration therapy. You have seen that if there is diarrhea, so doctors basically emphasize that there shouldn't be dehydration. Patient must be rehydrated. Patient must be given continuous fluid intake, so that the most dangerous thing will be dehydration. So it will be replaced with the lots of fluid. Antibiotics are of secondary importance. Antibiotics are then given to overcome the infection. The antibiotic recommended one is doxycycline. So thank you very much. This was about the cholera infection. And so you must go through this bacterial diseases, and if you have any type of query, you can ask during the live session. Thank you so much.